process used by plants and other organisms to convert light energy from the sun into chemical energy that fuels the organism's activities. The process always begins when energy from light is absorbed by proteins called photosynthetic reaction centers that contain chlorophylls, which are held in organelles called the chloroplast.
The photons of light energy are used in combination with carbon dioxide and water to create glucose, sugar, and oxygen. These photoautotrophs can create their own food through photosynthesis and release oxygen into the atmosphere to sustain aerobic life. The average rate energy captured by photosynthesis is huge, nearly 130 terawatts. That is 6 times larger than the net consumption of energy by all of humanity.
In plants and algae, photosynthesis takes place in organelles called chloroplasts. A typical plant cell contains about 10 to 100 chloroplasts. Within the membrane of the chloroplast is an aqueous fluid called the stroma. The stroma contains stacks, or grana, of thylakoids, which are the site of photosynthesis. The thylakoids are flattened discs bounded by a membrane with a lumen or thylakoid space within it. The site of photosynthesis is the thylakoid membrane, which contains integral and peripheral membrane protein complexes.
In the first part of photosynthesis, the light reactions, chlorophyll takes in photons and loses one electron. That electron is moved to pheophytin and then to a quinone molecule. This electron transport chain ends in the reduction of NADP to NADPH, a component in the second part of photosynthesis.
In plants, light-dependent reactions occur in the thylakoid membranes of the chloroplast and use light energy to synthesize ATP and NADPH. The light-dependent reaction has 2 forms, cyclic and non-cyclic. In the non-cyclic reaction, the photons are captured in the light-harvesting antenna complexes of photosystem II by chlorophyll and other accessory pigments. A second electron carrier accepts the electron, which again is passed down lowering energies of electron acceptors. The energy created by the electron acceptors is used to move hydrogen ions across the thylakoid membrane into the lumen. The electron is used to reduce the coenzyme NADP, which has functions in the light-dependent reaction. The cyclic reaction is similar to that of the non-cyclic, but differs in the form that it generates only ATP, and no reduced NADP is created. The cyclic reaction takes place only at photosystem I. Once the electron is displaced from the photosystem, the electron is passed down the electron acceptor molecules and returns to photosystem I.
Following the light reactions is the Calvin cycle, or the dark reactions. In this process, the enzyme Rubisco uses CO2 from the atmosphere and the NADPH formed from the light reactions to create 3 carbon sugars. This part of photosynthesis can take place in the dark, hence the dark reactions name. Light isn't needed because the light reactions uses photons, not the Calvin cycle.
Carbon fixation is a process in which carbon dioxide combines with a 5-carbon sugar, ribulose, and RUBP to yield 2 molecules of a 3-carbon compound, glycerate 3-phosphate, or GP. GP, in the presence of ATP and NADPH from the light-dependent stages, is reduced to G3P. This product is also referred to as pGAL, or even as triose phosphate. Triose is a 3-carbon sugar. Most of the G3P produced is used to regenerate RUBP so the process can continue.